So now we are back with another important scenario that we have to see in context of the photosynthetic process. This is a very brief topic, but is very important with respect to the photosynthetic process. And we would begin to look at how come the spongy mesophyll cells that we have in the structure of the leaf, they would contribute their role for the gaseous exchange. Now, whenever we use the word gaseous exchange, what we mean is that the carbon dioxide gas would go into the leaves and the oxygen gas would get out of the leaves. And this gaseous exchange would be happening differently during the daytime, and we must be very much clear that how come this concept or this phenomenon of the gaseous exchange would be accomplished with the help of these spongy mesophyll cells that we have got in case of the leaf. Now, if I were to quickly recall that how does the structure of the leaf it actually looks like. So you see at the top we have the cuticle, then here we have the paraepidermis, then we have got these palisade mesophyll cells, then here we have got the spongy mesophyll cells, and in them we have got these abundant air spaces. At the bottom we have the lower epidermis, and in the lower epidermis we have these guard cells, and in between the guard cells we have these openings coming, and these openings they are stomata. Now the situation is that when the light would hit the plants and when the light would start to enter into the leaf, so these palisade and the spongy mesophyll cells they would start to photosynthesize. And when they have started to photosynthesize, so they would be in the need of the carbon dioxide gas. And where is this carbon dioxide gas present? This is present in the external environment. So if I have to bring or if I have to take this carbon dioxide into the plants, then these stomata that I have got, they need to open up. So the thing that would happen to these guard cells is that they would become turgid. And the time they become turgid, so the CO2 it would diffuse into these air spaces, and the time when this comes into the air spaces, so it would go into the spongy mesophyll cells, it would be going into the palisade mesophyll cells. Now, since they have got the chloroplast in them, so the CO2 that has come here or that has entered into these air spaces, it would be rapidly used up. And when the CO2 would be used up, so this means that here the concentration of the CO2 gas that we have got in the air spaces this tend to decrease. On the other way around, these palisade mesophyll cells and the spongy mesophyll cells, when they would photosynthesize, so they would release the O2 into the air spaces. Okay, some of the O2 would be used up in their mitochondria, but excess of O2 would be coming into these air spaces. So as a result, the concentration of the oxygen gas would increase here in the air spaces during the daytime. So now there happens to exist a difference of the concentration gradient. By this, the concentration of the carbon dioxide gas that we have got in the external air, this would increase, and that of the O2 gas concentration that we have got, this tends to decrease. So as a result, the CO2 would diffuse into the air spaces of the spongy mesophyll cells, and the O2 would diffuse from the air spaces within the spongy mesophyll cells, and it would come into the external environment. So this is how the spongy mesophyll cells that we have here, they are contributing their role in terms of the gaseous exchange. And we at any cost need to take the CO2 into the inside of these leaf cells, the palisade and the spongy mesophyll cells, so that the rate of the photosynthesis could be carried out at the maximum rate.